Good morning. I hope you all are safe at home with your parents. While staying at your home, try to utilize the time fruitfully. Don't forget to follow our online classes time to time. I request you to all take homeopathic immune booster to prevent the COVID-19. Please stay safe at your home and avoid unnecessary outings. Don't forget to follow all the recommended preventive measures like hand washing, social distancing, etc. I hope you have already collected all my online notes and studied well. For a deep understanding of the subject, today I am delivering you a short video presentation for the better understanding of the online notes. You know, drug proving is an important topic. Drug proving is the foundation of our Materia Medica. One who conducts a proving will have a thorough knowledge in proving methodologies. From this topic, you will learn what is drug proving, how to conduct a proving, advantages and disadvantages of proving in both animal and human proving, etc. Okay, first let's know what is drug proving. Drug proving has been defined in aphorism 105 of Organon of Medicine as the process of acquiring knowledge of the instruments intended for the cure of natural disease. Actually, it is a systematic and orderly way of investigating the pathogenetic power of a medicine on a healthy person. By drug proving, we mean the positive effects of drug upon living organisms. That is, to know the pathogenetic effects of the drug. Drug proving are experiments with drug substances on healthy human being to note the disease producing powers known as curative powers of the particular drug. Now look into the glimpse of history of homeopathic drug proving. You know, Master Hahnemann presented before the world a collection of medicine substances and its pathogenetic action by a series of experiments conducted on his friends, his family, and himself. He published all these works in 1805. It is called Fragmenta de Verbis Medicamentorum Positivis Sive Insano Corpo Humano Observatis, which means fragmentary observation relative to the positive powers of medicines on human body. Later in the same year, he published his much acclaimed essay, The Medicines of Experience. In this essay, he details on how experiments with medicinal substances ought to be conducted in order to ascertain their pathogenetic effect. For drug proving, there are three essential requirements are needed. Let us see what are they. One is drug. In this case, the quality of drug must be pure. It must be free from all mixers with other drugs, and it must be possesses all its active properties. The other one is prover. In the prover, he must possess the proper balance in function and be in a normal, healthy state so that we can estimate the amount of the disturbance caused when we deliberately upset the balance of health. And lastly, environment, which means the surroundings of the prover must be those of his surroundings. So we must consider the environmental changes. These are the basic three essential things for the drug proving. Now, let us see what are the objects of drug proving. Here we should understand the pathogenetic effects, that is, disease producing effect upon healthy human being. Second thing is observation and recording of the alteration of the normal sensation by the production of the subjective symptoms. And then observe and note deviations in the appearance of changes in the tissue, organ, changes in the physiological function, etc. And finally, to produce the characteristics of the drug as they manifest in the disease states. Now, let's see about the selection of the prover. Actually, we depend two types of provers. One is animal and the other one is human. Basically, homeopathy depends solely on human proving. Now let us see what are the disadvantages and advantages of animal proving. The disadvantages are actually proving on lower animals should not be done because they cannot express themselves about their body and mental changes during the proving period. In this case, we lost the subjective and mental symptoms in animal provings. We will not get modalities and find sensation in animal provings. Not only that, the effect of some drugs on animal and humans are entirely different. Proper individualization is never possible on animal provings. The susceptibility and the idiosyncrasy is difficult to judge from animal proving. These are the disadvantages of the animal proving. But there are certain advantages are there in animal proving. They are, the drug can be proved in any quantity in animals. And at the same time, due to toxicological effects of drugs, it is not possible in human proving. And the other point is, the drug may be proved for any length of time without any limitations. But it is not possible in human proving due to fear of poisoning. In animals, we may go to the ultimate pathology and dare to lose the animal. In case of proving of unknown substances, we can give the drug for first-hand information without any fear. The examination of the physiological action of the drug can be done. These are the advantages of the animal proving. Now, come to human proving. In case of human proving, we should be very careful to selection of the prover. In the case of human proving on sick person is not at all allowed because the true effects of the medicine sometimes observed as symptoms of the natural disease. So we cannot ascertain the peculiar alteration of the medicine. In diseased state, the person may be hypersensitive or hyposensitive, and so the intensity of the symptoms may be increased or decreased accordingly. If accidentally the symptoms of the given drug are similar to the symptoms of the patient, he will be cured or partially relieved. And at the same time, if the drug used to proving is opposite in nature to his disease, it shall bring temporary relief. And if the drug is dissimilar to the disease, either there will be no effects or will form a complex disease. In case of proving on healthy human being, the best possible way of ascertaining the pure and the peculiar effects of medicines is to make a proving on healthy human beings in moderate doses. Now, let us see the advantages of the human proving. Except the negligible amount of disadvantages, human proving serves all purpose. They are subjective and mental symptoms with their modality and concomitance is observed in the human being. From the human proving, we get pure, accurate, pathogenetic effects of the medicine. Administration of medicine on healthy human being give response properly. And the peculiar characteristic of the drug, the rare, unusual, is only observed during human proving. These are the advantages of the human proving. Now, come to the qualities of a human prover. An ideal prover should have the qualities like the prover must possess sound health, free from disease, and individual can determine the effects of the medicines very distinctly. The prover should be intelligent, he should be delicate, irritable, and sensitive. He should be the lover of the truth, temperate in all respects of delicate feelings, who can who direct the minutest attention to his sensation. He must avoid all over exertion of mind and body, disturbing passion and distraction. Now, the question is, who is the best prover? The best prover is the healthy, unprejudiced, sensitive physician himself is the best prover. The idiosyncratic person is the best possible prover of the substances to which he is idiosyncratic. The next point is the medicine must be tested on different ages because physical appearance, anatomical development, and functions vary according to the age. We have to treat person of different age groups. So we must have a clear idea about the curative powers of medicines in different age group. There are diseases which are more common in certain age groups like measles, whooping cough, rheumatic fever are common in children, whereas enlarged prostate and cataracts are common in aged individuals. And also the susceptibility and physical irritability varies with age. So it is very important that we must prove the medicines on different age group. The medicines must be tested in many individuals because all the symptoms particular to a medicine do not appear in one person and not appeared alone at once and not appeared in the same experiment. Maybe some occur in one person chiefly at one time, others again during a second or the third trial. So, in order to get the whole pathogenetic power of a medicine, proving should be done on many different individuals. So let me conclude for today, and I will come again on tomorrow with the other part of principles of drug proving. Until then, goodbye. Thank you for your patience to listen me. Thank you.